Welcome to BIOL 100, Chapter 1, the study of life. So in this chapter, we're going over a little bit about what is biology, what is the scientific method, and talking about what is science really in general. So starting off, you are in a human biology course, so we obviously are concerned about biology, and that is the study of life, which so far is just restricted to planet Earth. And we'll go over what makes something a living thing in a little bit as well. So the science of biology. Again, biology is the study of life. So it's the study of living organisms and their interactions with one another and in their environments. Science, on the other hand, is kind of a way of knowing, right? It's defined as knowledge that covers general truths or the operation of general laws, especially when acquired and tested by the scientific method. So keep in mind that we do the scientific method to get to the science, a way of knowing, right? If we have a question about something, we go through the scientific method just to have those defined steps that include experiments as well as careful observation and reporting the results of those experiments. So some of the things that are in your book, we have these blue-green algae, the cyanobacteria over here on the left-hand side, and then we have stromatolites over here in Western Australia, just showing you how varied life can be here on Earth. We have everything from prokaryotic single-celled organisms all the way up to multicellular eukaryotic organisms or charismatic megafauna like large sharks or whales, elephants, things like that. And then we also have bacteria, right? Escherichia coli, E. coli, we've all heard about, I'm sure. This is a scanning electron micrograph, and we do have healthy-ish E. coli in our digestive tract, so everyone has it to some extent, but obviously we don't want to get too much of it or certain strands of E. coli, and that's when we run into issues. So as we get into the process of science, let's discuss what natural science actually is. So it includes fields of science related to the physical world and its phenomena and processes. As such, science and biology are restricted to natural explanations of phenomena, not supernatural. So when we're talking about God or creation, magic, space aliens, all of that, that is not science, that is supernatural. So that's not what we're discussing in this class. So now we get into scientific reasoning, which includes both inductive and deductive reasoning. So inductive reasoning is a form of logical thinking that uses related observations to arrive at a general conclusion. Then once you have that, you can kind of move into deductive reasoning, which is a form of logical thinking that uses a general principle or a law to forecast specific results. Okay, so with inductive reasoning, we have a number of observations and we usually draw some general conclusion. So let's say some observations if we're looking at a bunch of individuals that are in front of us, let's say on a prairie. We see that members of these species are not necessarily all the same and that individuals might be competing for resources, and the species are generally pretty well adapted to their environment, right? So we come up with the conclusion that individuals most adapted to their environment are more likely to survive and pass their traits to the next generation. So in this case, the conclusion that we get from our inductive reasoning, that can actually become the premise for our deductive reasoning. So deductive reasoning, we're usually operating off a general premise and predicting more specific results. So again, our general premise was our conclusion here that individuals most adapted are the ones that are surviving. So therefore, we would predict that if the average temperature in an ecosystem increases due to climate change, individuals better adapted to warmer temperatures will outcompete those that are not. So this isn't something that you necessarily need to be super well aware of. Just be aware in general that we have different reasoning within science. Now we get into how do scientists actually draw conclusions, which would be operating from the scientific method. But I would like to touch on here a little bit of what you might see in like the media, right? So we see all sorts of things in the media. Sorry, this is cut off. For instance, let's talk about coffee, right? Is coffee good for you or is coffee bad for you? Well, that depends on where you look. There are lots of scientific studies studying whether coffee is good for you, bad for you, whether it's the caffeine, what is it about coffee that could be linked to different things, right? Um, and if you read the news headlines, you might see something like coffee causes cancer, or coffee prevents some diseases, or caffeine can be poisonous. And all of these are kind of sensational headlines meant to grab your attention and make you read that paper. But the way that the media portrays science isn't necessarily always the actual science behind it, right? Coffee causes cancer. That might be a headline for a research article where they researched, you know, maybe they surveyed like 100 people that have cancer and 100 people that don't. And most of the people that have cancer drink coffee, and most of the people that don't have cancer don't drink coffee. That could be the cause of this headline, right? And does that mean that the coffee gave them cancer? No. And if you're only surveying 100 people on either side, that's not really a very large sample size to operate from. This is why we want a large sample size. This is why we want some diversity in our studies so that we can draw a more effective and probably more appropriate conclusion. So again, just some examples of things you might see in the media related to this coffee example, right? Like, why is coffee good for many Hispanics' health? 11 reasons you should drink coffee every day. A myriad of benefits. So it really depends. A lot of the questions that you might have for a scientist can really depend on who's writing it. And most of the time, scientific journals are really a great resource. And we'll go through that whole process here in a second. But be wary of what you read in the media. And especially if it's any media article really, but especially a decent media article really should cite what their source is. And if you're wondering, okay, is this sensationalized a little bit? Are they reaching on some of their conclusions in this media article? What you can do is you can go straight to the source and read that. And we will probably practice reading scientific papers in this class as well so that you feel comfortable finding and understanding them. Okay, so the way that scientists draw conclusions, so we use the best evidence available, right? And then we have to ask as readers, was the science properly performed? And a good way to tell that is if you see it in some reputable scientific journal, because that means that it's gone through something called peer review. Peer review is just like it sounds. As a scientist, you write up your experiment, your results, your conclusions, you put that into a paper, and then that is sent to multiple people in your field who should be able to say whether that science was properly performed or not, and whether they think this is something worth publishing. If they have issues with it, they might send it back, and then you can kind of put in your rebuttal and fix some of those issues. Or if it's not fixable, then you've got to go back and reperform the science, or you're not going to be able to publish it. But remember that science is a never ending process. And I think that this is really well illustrated by the COVID pandemic, right? When we first heard about COVID, we had no idea what it was, what it could do, and what we know about COVID changes almost daily. What we know about the vaccines changes almost daily. These things it's a never ending process. We are making the conclusion based on the best evidence that we have. And remember, that can be modified in the future. We might get better evidence tomorrow, and we might get better evidence in a year. So science is a never ending process. Science is never wrong or right necessarily. It's just changing. And that can be frustrating for some people, especially while you're working in a lab. But that's the part of science that can make it really fun and frustrating. Okay, so getting into the scientific method. So again, this is a series of well-defined steps, and we start off with an observation, right? So we observe something in the natural world, and that causes us to ask a question. And from that, we are going to form a hypothesis that might be able to answer that question. Remember, a hypothesis has to be testable. So it has to be something that we can, quote unquote, prove right or wrong. Do we ever prove a hypothesis? No. We can only support or not support or reject, is really what we usually say for not supporting. So after we form our hypothesis, then we make a prediction based on the hypothesis. So this is the kind of famous, when you're in 5th grade, you learned how to make a hypothesis based on if then, right? And I would argue the hypothesis is the if part and the then is the prediction. So let's go through a quick example. Let's say that we make an observation and we see, let's look at myna birds. And myna birds are always in pairs, right? So that's our first observation. And let's say that we see one of the pairs is always bigger than the other. This isn't necessarily true, guys. I'm just going through, let's say, an example. So I see all these myna bird pairs, and there's always one bird bigger than the other. So then I wonder, why is one bird bigger than the other? And my hypothesis could be something as simple as um, the male bird, or let's say the female bird, is bigger than the male bird. So I think in that pair there's a male and female, and I think the female bird is bigger than the male bird. So if this is true, my hypothesis that the female is larger than the male, what would my prediction be, right? My prediction would be that the female would be larger than the male, but my hypothesis should really be something behind that. So why do I think that the female would be larger than the male? And maybe it's for something like, if females use more energy for reproduction, they're taking in more calories, which means that females would be larger than males. So my hypothesis is the if part. If females are using more energy for reproduction, my hypothesis, then I predict that they would be larger than males, right? So now I'm going to test this, and I'm going to see, maybe I'm going to ban these birds and see which one's male, which one's a female. But I also want to test my hypothesis of, okay, how much energy does reproduction require? And that's kind of a hard thing to test, so this isn't the best example, but we're going to go through, we're going to design an experiment, we're going to test that prediction, and then we're going to go through and analyze the results. So was my hypothesis supported, or did I reject it and not support the hypothesis? Then I'm going to report my results, and if the hypothesis isn't supported, then I might try again. I might change it and say, okay, well, I don't think that females are larger because reproduction requires more energy. Maybe it's because they're foraging for food more than males are. Whatever it might be, we go back to the drawing board if we do not support our hypothesis. So now we go into the experiment, and to design an experiment, we have to make sure that it's designed really well and that we've really well defined our variables. A variable is something that can change whether we change it or it changes because of something we changed. Depends on what kind of variable it is. So the experimental group is the one that would experience an independent variable. So that's something that we're deliberately changing. Let's kind of turn our example maybe back to coffee. So let's say with coffee specifically, we want to study whether caffeine is good or bad for you. Maybe caffeine helps you focus more, right? So we're going to do a memory test with coffee, and we're going to see if the people that drink coffee with caffeine have better memory scores than they do if they're drinking coffee without caffeine. So this is kind of like what your first assignment also goes over, is defining these variables and understanding how the experiment's set up and what the results are and how you get better results. So again, independent variable is the one we're changing. So that's how much caffeine the groups are given, right? So it's the one we're changing in a systematic way. So we have both a placebo treatment, which means that we have a group that doesn't know whether they're caffeinated or not, but they're drinking coffee. And we don't want to tell them that they're placebo, right? Because then we get something called placebo effect, which I'm sure you've heard of. And placebo effect is essentially thinking that you are influencing your results in some way. So we're going to give this group decaffeinated coffee so that they don't know that we're testing specifically caffeine and that they don't know what's affecting them. So it's not part of our experiment, right? And then we also have a test treatment. This is our experimental group that's going to be getting the caffeinated coffee, okay? And so they're both getting it 30 minutes prior to the test. This is a control variable. So we want to make sure that as much as we can, other variables are controlled in the experiment so that we don't have any confounding variables. Confounding variable would be something that, let's say we gave this one at 45 minutes and this one at 30. Well, it could be a difference of 15 minutes. Maybe that changed something. So we want to make sure that the results we're getting are actually from the one thing that we're changing. This is why when you do an experiment, you normally change one thing at a time. Okay, so again, the control group is the one that's receiving a placebo, and that's just a fake treatment that mimics the experience of the experimental groups. That way they don't know, right? And then, sorry, I can't go back, but then we run that experiment and we make sure that we have all of our variables defined, and then we can actually report on the experiment. Okay. I also wanted to go over what a scientific theory is versus an everyday theory, because this is something that's kind of important because we use the word theory a lot, right? And in science, a theory specifically is something that hasn't been disproven. So you remember when I said our hypothesis is never proven because science is always changing, right? It could always end up being something different in the future. So we never prove a hypothesis, we only support or reject it. But in this case, we have everyday theories, which is an idea based on personal experience. So it could be something like, if I carry my umbrella with me, it's not going to rain, right? Or if I put valuables in the freezer, a robber's never going to find it, right? Those are your own kind of personal theories that aren't necessarily tested, or they might be tested, but it doesn't necessarily work for everyone. It's kind of based on your personal beliefs or experiences. A scientific theory, on the other hand, is one that hasn't been disproven. So all of the experiments leading up to it, usually thousands and thousands of experiments over lots of years, maybe 20, 30, 40, 100 years, whatever it is, they've never disproven. They've only supported this hypothesis. So these are theories like cell theory, which is that all living things are made of cells, general relativity, gravity, evolution. These are all things that have not been disproven by any scientific experiment. So these are what we refer to as scientific theories. We kind of accept them, pretty much as fact. But again, with science, maybe there's something that we find, some sort of cell that we find deep in the ocean somewhere, or some extreme environment that doesn't live up to this, and then we would have to change our theory. Is that likely at this point in time? Probably not, but we never know. Science is always changing, and we go with the best evidence we have. Science is also about finding different patterns, right? We observe patterns and we try to show a consistent relationship or link between those variables. That's what we often refer to as a correlation. Important thing about correlation is just because we see a correlation between two variables, it does not prove that one variable causes the other. This is the whole correlation does not equal causation. And just to give you a few examples, this is a study that was done in 2000 at a VA medical center here in Hawaii, actually, with 8,000 Japanese American men, and they were followed for 30 years. So again, with the coffee, we measured how much coffee they took in every day, and we compared that to their cumulative incidence of Parkinson's disease. So you can see here, the more coffee that they have correlates with lower incidence of Parkinson, and the people that drink less coffee correlate with higher incidence of Parkinson. So you can see the coffee intake here. Blue is the non-drinkers, and then we have different colors for how much. So this is the most coffee here. And the people that take in less coffee had a higher incidence of Parkinson's disease. Does that mean that coffee doesn't cause Parkinson's? We don't know, right? We don't know based on this. It could be, or it could be a link. That might be something worth studying. That's an observation we make from this study, but it doesn't necessarily mean that they are the cause of one another. Okay. Again, correlation not the same as causation. Here we see the temperature anomaly with CO2 concentration. Does increasing CO2 cause an increase in temperature, or does increase in temperature cause an increase in CO2? We don't know, right? That's something that's interesting. That's something that we're studying, something that we're looking into. But what we think we're seeing is that CO2 is causing increasing CO2 is causing many other things to happen, like increasing greenhouse gases, increasing sea surface temperatures, and then that is influencing the annual temperature. Um, Again, because two things have the same trend doesn't mean that one causes the other. Here's a funny one. As global temperature is increasing, so is U.S. postal charges. Does that mean that global warming, global temperature increases is causing our postage to go up? No, not really, right? It's probably just because we have inflation. So those two things aren't related whatsoever. And I think my favorite one, here we have our time down here. We have number of films that Nicolas Cage is in and number of swimming pool drownings that we saw that year. And you'll see that they correlate really well with each other. Do we think that people who see Nicolas Cage movies, or because Nicolas Cage comes out with a movie, that we have more drownings? Probably not. I'd like to think that he's not really causing that, but they are correlated. So just remember that just because there is a relationship between variables doesn't mean that they're causing one another. We can form a relationship between lots of different variables with different statistics. So be wary of things that you read and learn to question them and think about them in terms of the scientific method and whether you think that's valid or not. And that sometimes requires more research as well. Okay, so we're also talking about basic versus applied science. Basic science is also referred to as pure science sometimes, and it's seeking to expand knowledge regardless of short-term application of that knowledge. Applied science, on the other hand, is using science to solve immediate problems. These are problems defined by the researcher. So applied science might be something like finding a cure for cancer, whereas basic science is just understanding how that cancer got there in the first place, right? We just want to know about that so that maybe we can use it later for the applied science part. And again, we kind of went over this a little bit, but peer review, scientific progress is usually made through publication of peer-reviewed literature. And peer review, again, is a blind process where a scientist submits their experiment and other reviewers determine whether that has merit to go in the publication. And you'll see scientific papers throughout this course. They usually follow this format of having an abstract, which is basically like a quick summary telling you what this experiment is about, what it was testing, and what the results were. And then we have the introduction giving us some background, telling us what our hypothesis and predictions are, the methods, how we actually did the experiment, the results of the experiment, and then the discussion. So was the hypothesis supported? Was it rejected? Why do we think that happened? How does that relate to other research? And all of that other research that we're citing here is put into the literature cited or the references section at the very end. So if you are reading a paper and you're like, I know that this is peer reviewed, but it still doesn't really sound right to me, I'm not so sure, that's okay, right? It could be wrong. Science changes. And one good thing to do if you're having that thought while you're reading something is to go to the literature cited. See what other papers they cite and go back and read those papers. You might see something that changes your mind, or you might see something that validates what you're saying. So you'll see this in your book, but after Hurricane Irma struck the Caribbean and Florida in 2017, we saw thousands of these baby squirrels like this that were thrown from their nest, and they used applied science. So we knew how to rehabilitate the squirrel. So we had our basic science that we've done to maybe understand what things squirrels need, and then we used the applied science to actually rehabilitate the squirrels after that hurricane in 2017. So we'll switch gears a little bit now from the scientific process and start talking about properties of life. So again, this is something that could change in the future. As of right now, we define life as the following things. So life is carbon based because we've only found life on Earth, and everything on Earth is carbon based. And there has to be some order to it. In order to be alive, to be considered alive, we consider that you have to have sensitivity or be able to respond to stimuli. Okay. You have to be able to reproduce. You have to be able to grow and develop, regulate yourself, as well as homeostasis, which is just regulating your uh, internal body, basically, and regulating your internal temperature, internal pH, things like that, that basically keep your home safe and regulated the right way. You also have to be able to process energy. So this is kind of an interesting thing. We have these properties of life, and this can be challenging sometimes. For example, with viruses. Have you heard that viruses are alive? Are viruses alive? What do we think? Do they have order? Do they respond to stimuli? Do they reproduce? Do they grow and develop? Do they regulate? Do they have homeostasis? Do they process energy? So it depends on who you talk to, but generally, it's widely accepted that no, viruses are not alive. And a big part of it is one, all living things are made of cells, right? We kind of skimmed over that theory earlier. And viruses are actually not made of cells. Also, they do grow and develop, but can they do it without a host? They cannot do it without a host. So arguably we would say viruses are not alive based on these properties of life, and that they're not made of cells, which isn't on here, but it is something to consider as well. So again, order. Here's just an example of an organism, a toad, and it's a very organized structure, right? This guy's got lots of cells, he's got tissues, organs, and organ systems. So he has a very highly organized structure, and that's something that defines a living being. Again, response to stimuli. We actually have these plants here, the Mimosa pudica. These guys instantly droop and fold when you touch them. So they kind of like sink in real quick. And then after a few minutes, they return to normal. So that's a response to stimuli. That is, again, something that says that, hey, this thing is alive. Another response to stimuli here, let's see, can I play the video? Sorry, it looks like I will not be able to play the video. Oh, wait. Here we go. So again, something that makes sunflowers alive is that they respond to stimuli and they have solar tracking, right? If you would like to watch that video again, the link is down below, so you're able to do that. And then when we're talking about the order that we see, the organization that we see in living beings, it starts at the very smallest thing that we can possibly have, which is an atom, right? And then if we have a few atoms together, so let's say we have hydrogen atoms and our hydrogen atoms combine with an oxygen, we make a H2O or a water molecule, right? Then we have things that are macromolecules, which we'll go over actually next lecture. Um, and all of these things together can form organelles. So these are within your cells. These are things like your mitochondria, your nucleus. And then we have cells. We have different cells that go together and form tissues. We have different tissues that work together and form organs and organ systems. And then all of these things together create organisms. We have different organisms in populations, in communities, and then we have different populations and communities that form an ecosystem, right? And we have different ecosystems that are within our biosphere. So we work our way all the way from the smallest thing in the world to talking about large biospheres that include everything in between different ecosystems. Again, starting at the level of organization kind of that we normally start at, especially when we're talking about like animal or human biology, is we look at the organelles. So you can see the nucleus here that's dyed blue in these onion cells, this little tiny circle here. That's our nucleus. That's what contains DNA, right? And then we have cells. These are human blood cells. I believe these are red blood cells. We have tissues like human skin tissues. And then we have organs like our stomach, our intestine, our kidney. Um, uh, but stomach and intestine making up the digestive system, right? And then in a forest, we have all of these different pine trees. Those are each an organism. And together, all of those pine trees make up a population. And then things that are living in the pine trees or around the pine trees, all of those plant and animal species within this forest comprise a community. Okay. And within that community, if we have multiple communities coming together, we form an ecosystem, like this coastal ecosystem here in the southern United States, which includes living organisms and the environment. So ecosystems is both the environment and the living things within it. And the biospheres encompass all the ecosystems on Earth. Okay, and we talk about different biomes that we have, like a tundra biome or different wetlands or forests, and all of those are within this biosphere. And then, just to end all of this, talking about the diversity of life, we can look at things called phylogenetic trees. And you'll see these more, well, you'll see them a little bit in this class, but not too much. But basically, at the very bottom here, we have some common ancestor, right? So these are showing evolution normally. And here we're looking at the phylogenetic tree of life. We can see we are here where the star is. We're among the animals. But we can see this is constructed, and we have our separation of living organisms into three domains. So these are our three domains. We are within eukarya. So these are our kind of our more complex organisms, right? These are, for the most part, multicellular, complex organisms like animals, fungi, plants. Um, there are some other things that you'll see here, but for the most part, when we think of eukarya or eukaryotes, we think of animals, fungi, and plants. Then we have our prokaryotes. Prokaryotes include both of these orders here. We have bacteria as well as archaea. Okay, bacteria we've all heard of. Prokaryotic organisms within bacteria and archaea, for the most part, are single-celled or unicellular organisms, not very complex, do not have organelles. So like that nucleus we talked about, that's something that is only seen within the eukaryotic organisms. But these guys over here, again, simpler organisms, unicellular for the most part. Bacteria, archaea are generally more extreme. We see extremophiles within archaea for the most part. So we see these are organisms that can live in really extreme environments, like really salty, really low or really high pH, really high or low temperatures. That's these guys here. So they are still living, but they are a prokaryotic organism within the domain of archaea. So that's a little bit about a lot of different things, kind of introducing you to science, introducing you to the study of biology, and what we're going to see a little bit more this semester as we start to explore human biology.